Hello everyone, this is Dr. P. Mary Anupama, Head of the Department of Biochemistry, St. Joseph's College for Women. We'll be studying about structure of mitochondria in this video. The learning outcomes are, by the end of the course, the learners will be able to illustrate the importance of mitochondria in a cell, be able to describe the detailed structure and components of mitochondria, narrate the importance of the inner mitochondrial membrane. They'll be able to understand the localization of electron transport chain in mitochondria. The contents would be, first is introduction to mitochondria, then I'll be outlining the functions of mitochondria and a detailed structure of mitochondria and all the components of it will be illustrated in this video. Coming to introduction, singular is mitochondrion. We usually call it as mitochondrion. And if it is plural, when you are referring to multiple mitochondrion, then you use the term mitochondria. Where do you find these cell organelles? We find them in eukaryotes, like the animal cells, plant cells, and even in fungi. One special thing about these things is these are double membrane structures and they are used in aerobic respiration. You see that these mitochondria, they are involved in the generation of ATP, which is the chemical energy and the energy which is used by all our living cells. These were discovered by Albert von Kolliker in the year 1857, and he found them in the voluntary muscles of insects. And the term mitochondrion, it was coined by Carl Benda in the year 1898. We usually refer mitochondria as the powerhouses of the cell, and the nickname of this mitochondrion as the powerhouses of the cell, it is given by Philip Slikowitz, and this is in the year 1957. These mitochondria, they have a cross section of around 0.75 to 3.0 micrometer squares, and these are visible only upon staining and not visible to the naked eye. So the count also varies from cell to cell. Say for example RBC, they do not have any mitochondria, while the liver cells, which are very active, they have more than 2000 mitochondria in the liver cells. Yes, and these are compartmentalized organelles. And one more thing is they have their own genome, and this genome is just like the bacterial genome. The basic functions of mitochondria, first is we know that they are supplying the energy, they are the suppliers for cellular energy, and they are also involved in cell signaling, they are involved in cell differentiation, and at times they trigger even cell death, that is apoptosis, and they are very important for maintaining cell cycle and cell growth. Now let us see the details of the structure of a mitochondrion. Mitochondrion, it consists of 5 distinct components. First one is its outer membrane, that is the outer mitochondrial membrane. Then it has an intermembrane space, the space between the outer membrane and the inner membrane. Then comes the inner mitochondrial membrane. Then you have what are known as cristae. These are nothing but the infoldings of the inner membrane. And then finally the matrix, which is nothing but the space which is enclosed within the inner membrane, and that has the fluid. This is the diagram showing the detailed structure of mitochondria. So here you can see the outer membrane. So this outer membrane, it has some pores which we call them as the porins. Then you have the inner membrane. You see that there is a space which we call as the intermembrane space. Okay, and this is the inner membrane. The inner membrane, it is folded, which we call them as the cristae, while this fluid filled area, we call it as the matrix. This matrix, it comprises of lots of ribosomes and then DNA. Here you see small knob-like structures. They are the ATP synthases which are attached to the inner mitochondrial membrane, while these white ones are the granules. So we will see each of these things in the next upcoming slides. First, let us know about the outer membrane. So this is the outer membrane that actually encloses the organelle, and it contains large number of integral proteins. As we have seen previously, these proteins, we call them as the porins. So those porins, they are the transport proteins. And this outer membrane, it also has translocase protein that helps in the transport of large proteins, and this is through signaling which happens. And it also has enzymes, and these enzymes, they help in the elongation of fatty acids and also in the oxidation of epinephrine, in the degradation of tryptophan, etc. So you can understand that this outer membrane, it has lots of enzymes, and it also has proteins. If these proteins leak into the cytosol, this area would be the cytosol. So if the proteins of this membrane leak into the cytosol, it also induces apoptosis, which means cell death. So next to the outer membrane is our intermembrane space. This is located between the outer membrane and the inner membrane of the mitochondrium. This is also called perimitochondrial space. You see that the ion concentration of this area is same as that of the cytosol. So the outer membrane, as such, it is permeable to small molecules, allowing it to be in equilibrium with the cytosol. The protein which is localized in the intermembrane space, here it is the cytochrome C. It is attached to the inner membrane but located towards the intermembrane space, more into the intermembrane space. This is cytochrome C. Inner membrane. So this inner membrane of the mitochondria, it has three different types of proteins. First one is the proteins that are a part of this electron transport chain redox reactions. Then you have the ATP synthase, which is required for the synthesis of ATP. You also have many specific transport proteins that regulate the passage of metabolites into and out of the matrix of mitochondria. See, there are a total of 151 proteins that are associated with this, of which some are electron transport chain, some are a part of electron transport chain, while most of the proteins, they are like translocases, which are important for transport. The fourth component is the cristae. The inner mitochondrial membrane, it extends inwards as foldings. So numerous folds you will be able to see, and these folds are called as cristae. As such, these increase the surface area of the inner mitochondrial membrane, thereby increasing its ability to produce ATP via these ATP synthesis. And here the number of folds, they vary from cell to cell. This is based upon the demand of a particular cell. Say for example, muscle cells, if the mitochondria are present in the muscle cells, the number of cristae of that mitochondria will be more as compared to the other one. So here you can see that stud-like structures, they are present. Here these are the F1 particles which we are seeing. These are of ATP synthesis, involved in ATP synthesis. Next component is our matrix. So this matrix, it is enclosed by the inner mitochondrial membrane, and it comprises of 1/3 of the proteins that are present in the mitochondrion, and these aid in the synthesis of ATP. The matrix, it has very high concentrations of enzymes. You have mitochondrial ribosomes, you have tRNAs, you have the mitochondrial genome, which is a circular genome. And one more thing is, this matrix, it is responsible for oxidation of pyruvate, fatty acids, and also the citric acid cycle. The references that are used while making this lesson are, I referred to Principles of Biochemistry, Lineage's Principles of Biochemistry, then Biochemistry by U. Satyanarayana, and Harper's Illustrated Biochemistry. To summarize, what we have studied is the structure of mitochondria. Singular is mitochondrion, plural is mitochondria. The infrastructure comprises of 5 components: the outer membrane, which has 4 ends, 4-like structures. Second is the intermembrane space, the space between the outer membrane and the inner membrane. The inner membrane is the key one. It consists of all the proteins that are required for electron transport chain and even ATP synthesis that are required for the synthesis of ATP. It has several translocases. High amount of proteins will be there. Over 151 different types of polypeptides will be there associated with the inner membrane. Then you have the cristae, the foldings of the inner membrane. We call them as the cristae. Then the inner membrane, it holds the fluid, which you call it as the matrix. So this matrix, it consists of many proteins, enzymes that are required for the reactions to take place. It consists of genome, just like the bacterial genome, it will be there. That's why mitochondria are referred to as independent replicating units. Then it also has tRNAs, ribosomes, and one important thing is this inner membrane, it has stud-like structures which are facing the matrix. So those studs are nothing but the F1 particles of ATP synthesis. So thank you for watching this video. Do study the notes and then prepare for the assessment. Thank you.